everybody. Great, we got the recording on. Welcome to the inaugural Acer Cements Division's Rising Stars. This is a webinar series where we highlight rising stars talent that we want to highlight due to the excellence in cement and concrete research. And today we have Dr. Chelsea Heverin talking to us about advances in biocements. I would also like to highlight our two next speakers in this series. It is Dr. Kingsley Skillen from Texas A&M on optimizing sustainable concrete. So that's going to be in March 18th, 2 p.m. Eastern Time. And we also have Dr. Erica LaPlante talking to us about advances in magnesium oxide-based cements. And that is April 2nd, 1 p.m. Eastern Time. I also would like to remind everybody that we have the 14th Advancements in Cements-Based Materials. This is a meeting conference by the American Ceramic Society Cements Division. That is going to be June 19th to 21st at Missouri S&T in Rolla, Missouri. And it's a great conference. This is the premier conference for scientific advancements in cement and concrete research. And it is one of my favorite conferences. So I am obviously very, very biased, but I hope to see all of you there. And abstract submissions has opened. So those are due April 1st. So hopefully seeing some of your abstracts coming in for that. All right, so to introduce our wonderful speaker, this is a dear colleague of mine and friend, Dr. Chelsea Heverin. Dr. Chelsea Heverin is an assistant professor in the Mechanical Industrial Engineering Department at Montana State University. She earned her PhD in mechanical engineering from the University of Colorado Boulder, where her dissertation focused on the impacts of aging and kidney disease on bone fragility. So she can talk to you all about bones, and it's amazing. I learned a lot. Dr. Heverin did a postdoctoral training in civil engineering, also at the University of Colorado Boulder, and engineered photosynthetic living building materials. Her current research focus continues to work towards the understanding of determinants in bone fracture resistance and applying those lessons towards bio-inspired engineered living materials. So we're very, very excited to learn about the latest advancements in this field. And in 2020, something that we did together was really, really cool. We won the an Emeritus Award for the National Science Foundation idea. Oh, it's right there.
It's like right here. Oh, it's like, and the screen is having hard. There it is. Okay, good.
There it is. The National Science Foundation idea machine. So her research has been focused, has been featured in the New York Times, ABC, Yellowstone Public Radio, and I am just so excited to hear more about it today. So with that introduction, I'll turn it over to Dr. Chelsea Heverin. Thank you for being here.
Thank you so much for the amazing introduction, JP. It's my honor to be here. I'm so excited. I'm just going to share my screen. Can you all see my screen?
Yes.
Wonderful. Okay, I'm going to kick it off here. So again, it's an honor to be here. This is just, it's so exciting for me to get to connect with the cement and concrete material community and discuss something that's probably a little bit different, which is learning from bone to engineer tomorrow's more sustainable biocemented materials. So I don't have conflicts to disclose, so I'll just jump right in here. So JP already introduced me, so I'm not going to spend too much time on this, but my background's a little bit different for someone who's interested in construction and infrastructure materials. I come out of a background where I studied bone. And the reason I want to mention that again is that, you know, near the end of my PhD, after spending years thinking about why bone breaks, I started to flip the problem and I started to be really interested in why bone doesn't break for so many people for so many decades. And that's actually something profound, given that bone is made of mostly lightweight constituents and stiffened by calcium phosphate biominerals. And I was interested whether there is a design space that we could pursue to improve the sustainability of manufacturing other materials that we use every day around us. And so in working with Will Schrubar doing a postdoc, we started working with living building materials that were photosynthetic that we'll talk a little bit more about today. But I'm happy to say that in the last few years up here at Montana State University, we've been able to merge these ideas and think about how we can use microbial biocementation and other processes to approach how we build in a different way. So I wanted to mention right away that the work that I'm showing you here is, for the most part, the product of a wonderful team of graduate and undergraduate students, and I'll call out a few of them along the way. So my lab works on two main things. And the first we're mostly not going to talk about today, which is about how bone cells interact with the material around them. I promise you it is, for the most part, not a lecture about bones, but we'll have to talk about them a little bit. But the reason we care about that is that the more we learn about how bone cells can modify a dense, mineralized environment around them for decades, and heal fractures and otherwise promote fracture resistance, the more that we can design our next generation of more material system of more sustainable material systems. And so on this lower row, what I'm representing here is that the major research thrust of my lab right now is actually engineering sustainable building materials where we use microorganisms or other biological processes at low temperatures to help us build or modify materials. Okay, so what are we working towards here? Like many of you that are here today, we probably have the common interest of thinking outside the box about how to improve the sustainability of making construction or infrastructure materials. I am motivated in part from the intensive time, energy, and labor resources that go into manufacturing cement. Of course, I don't need to tell this community about how cement manufacturing has an outsized carbon footprint. And it's, of course, such a ubiquitous material. So we need more sustainable options for manufacturing some of these materials. Now, I'm not going to tell you today that we need to be making everything around us using microbes. Absolutely not. But instead, that using microbial biocementation and a few other processes, we can expand the toolbox and bring in more sustainable approaches for some applications in this direction. I'm sure many attendees here are working on other extremely interesting alternative directions to the same problem. Okay, so let's jump in with microbes as builders. So we are motivated to utilize microbiology to help us build, but microbes are building already. And sometimes you have to zoom in and look at the natural environment around you to see this, and then think about how you can use it. So Sporus arsena pasteuri, which here we see in association with calcium carbonate, is a native soil microbe, one of many, 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 many that can promote mineralization near it. Some microbes can produce biomineral intracellularly. This microbe and others produce biomineral extracellularly because of their metabolism. And that can lead to these locally densified structures. So one interesting thing is that if you were in a cave or a different environment that has a lot of limestone or limestone-like geologic formations, if you took a little piece of that, and I'm not suggesting you go to your local cave and break off a stalactite, please don't do that. But if you were to look inside one of these pieces, it would be really common to see bacterial casts. So oftentimes microbes are actually ubiquitous in their association with biomineral precipitation in nature. So there are many different metabolisms that can be useful for bacterial biomineralization. One that we and others use a lot is called ureolysis. So here, the microbe makes an enzyme called urease, and when it comes in contact with urea, then you get this hydrolysis of the urea such that the outputs increase the alkalinity of the local environment to that cell and increase the supply of bicarbonate. If there is a calcium source available, you can have the precipitation of calcium carbonate, which gives us, again, limestone-like features. Okay, so how do we use this? This has been used for some time, especially where I am now at the MSU Center for Biofilm Engineering and other places too, but this has been used for a while. And oftentimes this microbial-induced calcium carbonate precipitation, which we'll call MICP, is very often used to bridge together aggregate. And we'll talk about some applications of that in a second. But what you're looking on the left is a schematic of sand grains that have these little bridges, these little necks of this stiff, brittle calcium carbonate in between them. On the right, we're looking at a micrograph of these polycrystalline bridges that can, again, that can hold our sand grains together and in some cases coat them. So how is this used today? So MICP has been used for many different kinds of applications. So some of the applications that are used in the Center for Biofilm Engineering up here range from dust suppression to soil stabilization because of coarsening soils using these biocementation processes or sealing cracks. Some of this is used at the field scale. So there are technologies that have come out of this center and others that, for example, seal leaking oil and gas wells. So that's actually commercialized and hundreds of wells have been sealed this way, so on and so forth. And so it is a scalable technology and it has good usage in many applications. Relatively recently, computers went a little slow here. There we go. Relatively recently, our lab and others, quite a few others, have started to ask, could we use this process to construct things that are useful? So useful building and infrastructure materials. This is a low temperature process. We don't we don't go very much above room temperature actually when interacting with microbes in this strategy, which gives us some unique ideas about how and when and where we can build or seal a crack, etc. So there are now commercially available building materials that are constructed using MICP, and I'd like to highlight a couple of them before we talk about how we might be able to move forward with continuing to improve this sort of strategy for manufacturing materials. So Biomason is a company that produces a paver tile, and it's essentially crushed granite that is biocemented together with microbially produced biocement. And they use ureolytic microorganisms such as Sporosarsina pasteurai and others. And on the right, what we're looking at are a little bit more sophisticated geometries that are being produced by Prometheus materials, which is exciting because this has a connection to the project that we were working on it, several years ago back at the University of Colorado. And here, instead of using ureolytic microorganisms, the biocement is produced in association with photosynthetic algae. And some of these blocks in the right are actually developing pretty decent strength. So these are commercially available products. They are not ubiquitous. We're not ready to build a lot of the world around us with these kinds of materials, but we're starting to be able to do more and more. I would argue, though, that building with microbes has the potential for important and broad sustainability impacts, but we're not yet reaching the potential of these spaces. And a few of the challenges that, if we were to surmount, we could increase what we can do with these microbes and biocementation as a process are as follows. The first is increasing the strength of these materials. By and large, bacteria biocemented materials are not yet that strong. Some of them are better than others, but we have not hit a ceiling yet for how strong they can be. And there are a number of ways we can potentially improve that strength, and we'll talk about some of them in a few minutes. Another is increasing the viability of cells within those structures. Now, some of you are probably thinking, oh no, I don't want that. I want microbes to help me build something, and then thank you very much, they can be dead. I don't want more microbes. That's fine. But as I'll hopefully be able to argue effectively to you, if you are able to keep cells alive, you may be able to confer some very unique functionalities to your materials that might enable transformative advances to how we make and use things around us. Okay, so we're going to be talking about bone and how that can help us surmount some of these challenges. And I'm starting with a picture of Muscle Guy trying to destroy this cow femur because I want to impress upon you that bone is really hard to break. Bone is fairly lightweight. It has great properties. It assembles at low temperatures, and it is really, really high performing for what it's made of. And we would like to move closer to that space. When I say that bone has decent properties, let's talk about it. So if we look at the fracture toughness on the y-axis and Young's modulus on the x-axis, we can see that our ceramics have high modulus and kind of mediocre toughness or even low toughness. Our metals are in this great category in the upper right where our biological materials generally don't live. And bone has pretty high toughness for its modulus. It's stiff enough to get the job done, and the fracture toughness is quite good. Cement would have it's a little stiffer than bone, but is not as tough. Um, by contrast, if we look at our current biocemented materials, most of the current biocemented materials have decent toughness and have way too low of moduli for the kinds of applications that we might like to envision using them. There is a lot of opportunity here to move to the right with this system. Okay, so let's talk a little bit about what bone is. So I can share some ideas about how we might draw some inspiration from it in improving how we work with biocementation. So bone starts with a template of mineralized collagen. So it is a scaffolded material, and the scaffolded collagen material is stiffened by hydroxyapatite. So bone mineral is not calcium carbonate. It is calcium phosphate, and these are nature's smallest crystals. Bone is about 65% mineral, and the rest of it's organic. It's actually impressively hydrated as well. So fibrils of calcium and mineral assemble as fibers. These fibers are further stiffened by crosslinks, and these mineralized fibers pack in these oriented sheets. And these sheets roll up in osteons within the dense part of cortical bone. And what that looks like is that you almost have these sheets of twisted plywood. And so bone has some incredible microarchitecture to it. And the importance of it is that, remember how tough bone is? Well, imagine a crack starts on the left and tries to go to the right of this structure with so many interfaces. Much of bone's extrinsic toughness is because of this microstructure. So let's talk a little bit about what bone can do and contrast it with what our building materials right now can't do. And I can, again, hopefully convince you why we're trying to draw inspiration from these systems. Let's say I have this cute little house, and after a while I decide it's too little and I want to build another floor. So I want to build an attic and increase the load on the foundation of this building. There's not much that can be done. That foundation anyway isn't going to sense that there's a greater load and make itself bigger. There's no way that that happens. We'd have to intervene. Bone, by contrast, does something totally different. What you're looking at here, these are cross sections of a tibia from a mouse. And the labels, the green and the red, are fluorescent dye that was injected into the animal. And what you're seeing here, this negative 1,000 microstrain up to negative 2,200 microstrain, this is from intentionally loading the tibia. So the negative 2,200 is a big enough strain on that tibia on the live animal to cause the bone to grow. So the green and the red show you where the bone is growing. It is adapting to an increased load. That's possible because bone is filled with cells. Now imagine also that we have a brick wall and we want to replace one brick and not the whole thing. Well, bone does that too. And so what you're seeing in green here is a place where bone recently took away some material and put back new material. For those of you who are, maybe 30 or younger, you're going to have a totally new femur in 5 years. It is phenomenal, and you won't know when or how that happened. It just happens a little bit, little brick by brick along the way. Okay, this happens because of bone cells. There will not be a quiz on bone biology. Don't worry. I just want to introduce a couple players here. Osteoclasts take bone away. Osteoblasts put it back. And osteocytes engineer the entire thing. They tell everybody where to go. But when you look at these networks, even in compact bone, you can see how porous bone is and how fluid has the potential to move through this structure. It can move through these little pores. Here are these osteocytes embedded in these network of cannuliculi. And those osteocytes are strain sensitive. They're the ones that send signals to other cells when they feel that loads are exceeded. They also send messages to other cells about when and where to mineralize. So our first research question here is, could we improve the efficiency and precision of biocementation by better control of microorganisms? Maybe not to the degree of exquisiteness that bone can achieve, but can we improve this? And the answer is, yeah, I think so. So again, trying to keep this analogy with bone science going, bone cells know where to go because of chemical signaling from mechanosensitive osteocytes. They get directions from other cells. And then they attach to surfaces through physical and chemical interaction. So could we utilize similar processes as this to improve spatial temporal control of microbial biomineralization? as opposed to this random biocementation of something like sand? The answer is yes, we can do this. So we're working on a few ways to do this, but one that showed good success early on was from functionalizing our sand surfaces with essentially silane coupling agents. So we functionalized sand surfaces with 3-aminopropyl methyl dioxysilane, or APMDES. Won't be saying that too much. And this creates an electrostatic interaction between negatively charged cell surfaces, that's the bacteria, and positively charged sand. And that's important because sand is by itself normally negatively charged, and that doesn't promote great interactions between these things. In bone, cells have one charge, and biomineral has the opposite charge, which improves biomineralization. So when we functionalize these surfaces, so on the top row, you have no treatment, and on the bottom row, you have treatment. The left column is confocal microscopy, where red and green show you where cells are. And on the right-hand side, we have scanning electron microscope images. So with no treatment, that means we're biocementing the sand, but it didn't have the surface treatment first. We don't see very many microbes at 15 minutes after we put the sand in a biomineralizing solution. And that makes sense. That's really fast for this. When we treat the surfaces, we have this immediate attraction and almost a monolayer of cells over the surface. They don't always survive, but they're stuck to the surface and their urease is available. We find that these microbes then create this really nice scaffold, and the scaffold of literally cells, we'll be talking about scaffolds later, helps promote this biomineralization of the structure. What this gets us is that when we have, let's say, untreated cubes and prisms, they're a little bit coarse, even after 7 injections. They have okay strength, but they're not very pretty. But with our treated conditions, we're able to build nicer things in the same amount of time. And we see that reflected in our strength gain as well. So after 3 injections, our strength gain is really considerably higher than for our untreated specimens. And it takes those untreated specimens a long time, out towards 7 injections, to catch up. Importantly, the results are not just described by how much calcium was in the structure, which tells us that there's other attributes at play too. One thing that we found that I found really interesting is that these calcium carbonate bridges, here seen in green on our SEM EDX map, have a different shape and size with our treatment. So we think there are changes to how biomineral nucleates and growth on our treated surfaces, and we're working to understand that more. Okay, so what's next for us in this space? Well, we're trying to, again, improve how we pattern microbes on surfaces so we have better control over where we biomineralize and where we don't. So we're adapting strategies that we've been able to use to stick cells in patterns on surfaces, such as these generated at Montana State University, to pursue that question. So another direction that we're working on here is to try to control the external microarchitecture of biomineralized materials. So it's a little bit less random and a little bit more towards desirable geometries that we want. Again, bone is made of lightweight, not very strong materials, so it has to be architected really well in order to be as strong and as tough as it is. As a second question that I'll just tease you with, what if these scaffolds were alive? Because wouldn't that give us some interesting design possibilities? So let's see what we can do in that space. So let's start with our first foray into these scaffolded living materials. That was during my postdoc, and we were working with photosynthetic microorganisms. So we were able to put these photosynthetic microorganisms into kind of a hydrogel scaffold, which they then biomineralized. And it was good for building things quickly. It was not as good for some other things. The hydrogel was not as robust to some systems. And we couldn't control, let's say, a lot of microarchitecture features about that hydrogel scaffold. It worked pretty well for us for what we wanted to do, though, and it was very exciting that we were able to keep cells alive in some conditions for a month. So if we kept the scaffolds humid, they lived for a long time. Moving forward at Montana State, we've wondered if we can take other strategies here. So one of our strategies is to use an entirely different sort of biological scaffold. And a candidate that we were interested very early was using fungal mycelium as a scaffold. And this is not random. This is because many species of fungus grow really quickly, and they can have their own biological activity. So species like Neurospora crassa produce urease. remember that our biomineralizing microbes produce urease, so maybe it could mineralize itself. We wanted to know if that could be something that a living scaffold is capable of, or if it could support the biomineralization by bacteria, or if we could shape this scaffold to be a useful microarchitecture. So we performed an experiment where we grew up en crassa, and then we had 4 conditions. Two of our conditions, we kept the fungal mycelium alive, and we fed it different things. The first was a rich nutrient source, and the second was a defined nutrient source, but the organism is forced to do urea hydrolysis. So we thought that would probably lead to greater precipitation of calcium carbonate because this microorganism has very diverse metabolism. We also did another condition where we would kill the mycelium and then we would grow up ureolytic microbes as pasteuri on this mycelium, and then we had an abiotic mineralization and control. So what we saw from this, so as a guide to these kinds of graphs, so we have time on the x-axis, and with calcium and urea, when you're performing biocementation, if these values go down over time, it gives you a sense that more biomineral is being deposited. And whether it's in association with urea hydrolysis or not. So what we can see is that for these BICP conditions, so that means the bacteria conditions, so they're scaffolded and planktonic. Planktonic means bacteria by themselves, no scaffolds. In either case, when we have the bacteria, the urea and the calcium are consumed very, very quickly. Nonetheless, we do have urea and calcium consumed, albeit more modestly, by the fungus itself, but it does take its time. We do see a big difference between when we use the defined media versus the malt media. When we then go look at the dried mass, we see that by and far the highest dried mass is when we use the bacteria on a scaffold. As a percentage of how much mass we grow, it's actually similar between some of our fungus conditions and the bacteria conditions. It's just that we can build a way bigger scaffold first in the malt media and then kill it and then mineralize it with bacteria, and that helps us build the fastest. And so we saw that as an attractive way to move forward. Furthermore, when we go look at what we built with fungal biomineralization, when it mineralizes itself, there's plenty of mineral, but it's mostly right on the fungal hyphae. When we mineralize the thing with bacteria, we get this beautiful, again, red here is the calcium-containing calcium carbonate mineral, this mineral that bridges together the scaffold, and that's what we want to see. I throw this image in for fun, but I think it's beautiful. What we have here is a fungal hyphal tip with a single crystal of calcium carbonate with 2.5 bacterial casts on it, and it really shows you the spatial association between the microbes and what they're producing. It was exciting to us that our scaffold stayed alive for a long time, like very robustly alive. So here we asked if living scaffolds could stay alive if we mineralized them, and the answer was absolutely. We measured this by basically we let it dry for 2 or 4 weeks at room temperature or 30 C, and then we give it new food and see if it grows. And so we measure that by the percentage of a plate that it grows over, and so everything grew. So our living scaffolds are able to stay very, very robustly alive for a long time. We also asked whether the bacteria would stay alive this way, if they like to stay alive when they're on en crassa scaffolds, and the answer is yes. So when we look at, so in this key here, S is scaffold, P is planktonic, so that means no scaffold, plus is mineralized, and minus is not mineralized. So on the left, we see that when we have planktonic conditions, so no scaffold, if we dry planktonic cells for 2 or 4 weeks, they're dying. By 4 weeks, they're dead. That makes sense. When they're on a scaffold, they're not dead. Now, some of you who might work with microbes might think, yeah, but it's not necessarily those microbes that are living on this nutrient-rich food source for a month at very pleasant temperatures. And that's true. And so then we do urea hydrolysis assays. And again, the lower the number here, the more urea it's hydrolyzing. That's a good thing if you want to see urea hydrolysis. And we see that even on our 28-day conditions, when there's a scaffold, there's a lot of urea hydrolysis after a month. And so, and that much less so at 30 C, but at room temperature, that's pretty good. Compared to needing to store these in special conditions, these are exciting findings for being able to keep cells alive for a long time. We think those ureolytic microbes really like to live on the fungal mycelium scaffold, and it helps protect them from environmental stresses. So where are we going from this? I was interested in whether we could start using fungal mycelium to control the internal microarchitecture of biocemented materials. This is very new data, but the answer is at first pass, it seems like yes. So you recall those osteonal type designs that we've been using. So we've been able to use fungal mycelium and generate osteon-like biomineralized structures within materials with sand. This is one of the first ones we made. It's basically a micro CT image of a prism that we later broke during flexural testing. We've made new ones of these with a much higher osteonal density, but those are just recently made, so we don't have images of them in here yet. But you can see here on image C, we've got sand grains. You can see what biocementation looks like through the sand grains. You have these little bridges that are incomplete, but you have much higher, much bigger calcium biominerals that are accumulating within this in this case, like a little ring of that fungal scaffold that forms the osteon, those concentric rings. So we see that with this pink calcium biomineral here. So we think this is an exciting space, and we're really excited to explore how we could start controlling the microarchitecture of these kinds of materials through biology. So some key takeaways for you here. First of all, we found that biotrapping cells enabled a faster, more efficient biocementation that was also more precise. We're trying to push that forward and be able to do more with it. We find that using scaffolds is very helpful for biocementation. I'm not surprised. In nature, most materials that are biomineralized are scaffolded. We are finding that bioscaffolds such as fungal mycelium can be a good option. So we find that it gives us a bigger toolbox that we can engineer these materials with. And some of the tools here are very exciting to us to give us control over things like microarchitecture. And again, I will contend that bone has many lessons for designing biocemented materials and pushing what they can do. I don't think that the next generation of biocemented material needs to be fully biomimetic. There's not a need for that. But when challenged with difficult design spaces, bone has made it work. And so we can look towards that system for a lot of inspiration. With that, I want to say thank you so much to the organizers, and particularly to JP for the invite. This has been really fun. I'd like to thank my students, collaborators, funding, and just throw it out there that our lab is hiring PhDs and potentially postdocs. And please reach out if you have if you would like to discuss some of this. But if there's time, I'd be happy to take questions. Thank you so much.
Yeah. Thank you so much, Chelsea. Just doing a virtual clap for this awesome presentation you just gave us. And everybody, I did forget, please do put your questions in the questions and answers box down below. And while you do that, I do have a couple of questions for you, Chelsea. The first one, really easy. Can you please tell us a little bit about what you think these immediate applications are for biocements? What are the most immediate, and maybe what are some of the ones that are maybe coming in the future?
Yeah, so right now, you can go find, again, commercial wells that have been sealed using biocement that exist, that have been produced with thousands and thousands and thousands of gallons of biomineralizing solution and microbes. So those are non-hypothetical, they exist, it is commercial. As I mentioned earlier in the presentation, there are companies such as Prometheus, such as Biomason, who are making certain materials that in some instances can perform tasks that used to be performed with cementitious materials. And so those are non-hypothetical as well. As far as where this goes, I'm really excited as some of these systems start showing higher strength, where we can replace more types of materials where cement has had a role. As a bone person, down the road, I'd be really, really excited if more of these systems were included living cells and could either tell you something about your environment, such as acting like a biosensor, or it could help participate in something like crack remediation. Right? That's been really hard to do in concrete type systems. That's a really harsh environment for cells. These systems are a much less harsh environment for cells.
I think that's such an interesting and exciting direction with biosensing, and specifically looking at some of the durability issues that we see in concrete materials. So that's fascinating. We do have a couple of questions here coming in, so keep this coming. Okay, so the first one for you here, Chelsea, is from Will Shubar, Dr. Will Shubar. Can you elaborate more on how these living scaffolds are keeping the biomineralization microbes alive? Is it providing moisture? And if so, is there a limit to these nutrients or both?
That's a great question, Will. Thank you for the question. So first of all, to clarify, uh, our viability work with the microbes with pasteurized is completed for situations where the N crossus scaffold is not alive, but the microbes have we've inoculated those scaffolds with pasteurized and they stay abundantly alive. Those scaffolds reach an equilibrium mass far before the end of that 4 weeks. They reach it quite quickly because we drive off water at, for example, 30°C drying conditions rather quickly. My suspicion is that, and I can't say this with full confidence yet, we're working on this right now, but my suspicion from some of our imaging work is that the microbes find safe harbor within the mycelium. We know that they can access the interior of the mycelium. I also suspect, but again, working to confirm it, that this autoclaved mycelium is an excellent nutrition source for both carbon and nitrogen for pasture. So that's what we're working to explore. We were very surprised how viable pasture was in that situation, including at the high temperatures.
That is fascinating. Um, Other question. This is coming from University of Miami, Dr. Pranoy Surneni. Um, Pranoy asks, what are the challenges for scaling biocements, bioaggregates, and other products, including with respect to ASTM specifications?
That is a really good question and a timely question too. There are many challenges. So again, some of the well sealing work has, in my opinion, paved the way for working with the type of organisms that we work with here, such as pasteurized. Are some of my collaborators at the Center for Biofilm Engineering have been able to show that it's reasonable to work with microbes like that in non-sterile conditions and grow sufficient quantities of them using cheap resources to surmount some of these scalability issues for some of the applications of biocementation. In other instances, it's not so clear. So we have not yet tried to scale up fungal manufacturing. That has probably different considerations. Our system does not involve synthetic biology, but sometimes biocementation does. In some systems it does, and that has lots of challenges for scale up. But yeah, that's important. I love the question about the basically the regulatory question of how should we test these things and to what standard to which that they should apply. And these questions are starting to emerge in this space, which I'm really happy to see. It's not figured out yet, but people are talking about it. And that's important. If we're going to be using these as common materials, we need to make sure that they perform in a way that we expect and are reliable.
Absolutely. I
Yeah.
agree. One more question. Um, so, can you paint us a picture for what functional groups in the surface of, let's say, sand are being utilized by bacteria to precipitate carbonates in the case of microbial induced carbonate precipitation? Are there some functional groups that are most useful than others? What have you seen?
Also an interesting question because sometimes these studies are done on really super clean sand and sometimes they're not. And these processes are used on a wide variety of soils. And so the answer is it depends, which is not very satisfying, but I can get into a little bit here. In our situation, for calcium carbonate, excuse me, for MICP as a general case for bridging together sand, those microbes do not have to interact with any functional groups on the surface. You can have the microbes form biofilm and attach to the surface, and then the calcium carbonate precipitation occurs because they have access to urea, which they break down through urea hydrolysis and change the solution chemistry around themselves to be locally alkaline. And so if bicarbonate is available, if calcium is available, you get calcium carbonate. And so when we watch this, we've started to do live cell imaging to watch this, which is really fun. You see these biominerals nucleate really close to the organisms, and quickly fill space and form these bridges, which is fun, right? I'm not going to say that they can't ever interact with stuff on sand, because real sand has all sorts of stuff. There's iron containing things. There's lots of metals that show up on sands, right? And so it's depending on the microorganism. There are ways where they can utilize those other um, constituents.
Right. Yeah. I mean, clean sand versus clays attached to the sand. I can just imagine the variety of
Totally.
phases dealing with in the surface chemistry diversity.
Yes. Yeah. And so we're starting to really pay a lot of attention to that as we start to try to pattern these surfaces. And when we start to work with real sands, as you'd expect, it gets more complicated.
Yeah,
And that's important
absolutely.
if you want to scale this up.
Right. It all comes full circle.
It
Well,
does.
we'll we'll stop it there. And thank you so much for the awesome talk, Dr. Chelsea Heverin. This was very inspiring. And thank you, everybody, for attending and participating. We hope to see you in the next Acer's Rising Star webinar series in about 11 days. So thank you so much. And I'm seeing few comments here to thank you, Chelsea, for all your time and the wonderful talk. So, no,
Thank you so
thank you,
much.
everybody.
I really appreciate it. Take care.
All right. Thank you, everybody. See you later.